RNA encryption. Maybe. We're going to be looking at a news release and then the article that underlies that news release. The news release is entitled Encrypted Messages in Biological Process. It's from the Department of Molecular Biology and Genetics of Aarhus University. And that, by the way, is in Denmark. I think this is the molecular and biological, molecular biology and genetics department. And so you can see it's obviously not totally English, but they speak mostly English in Denmark anyway, so. The blurb starts with RNA modifications can encrypt the RNA code. Encrypt is an interesting word to use in this context, and are responsible for a very sophisticated control of RNA function. A Danish-German research team has showed that modified RNA bases have a great impact on the dynamics of gene expression from DNA to functional RNA. The study yields important new insights into how the basis of RNA modifications can affect the function of mature RNA molecules. The genetic material, DNA, is located in the cell nucleus where gene expression is controlled. So far this is kind of standard. DNA is copied into the less stable RNA for translation into protein in the cytoplasm, messenger RNA or protein coding RNA, or for mediating independent functions as non-coding RNA. RNA is processed through several maturation steps to ensure its proper expression and localization. One of these maturation steps is called splicing. Maybe localization is part of what they're thinking about, I'm not sure. The non-functional introns are excised from the newly made RNA in the splicing process to build a mature and functional RNA consisting of exons only. And figure 2 is just illustration of this kind of thing where there'll be an exon, an intron, and another exon. In real life there might be 5 or 6 or 10 of these things. And you cut the intron out at certain specific spots, and you paste the 2 exons together and they become the actual RNA that you want, and the intron is left to float around and be degraded or whatever. Now, we'll come back to this M6A in a little bit. RNA is composed of 4 bases, abbreviated A, U, G, and C, corresponding to the DNA bases, which are A, T, G, and C. T is thymine, which is simply 5-methyl-uracil. Then is thereby disseminating its message in a fairly simple code. In recent years, research has shown an unprecedented impact of RNA modifications at all steps of the maturation process. And we're going to go through at least a list of how many there are. More than 100 RNA modifications have been identified, with roles in both inhibiting and facilitating binding to proteins, DNA, and other RNA molecules. This encryption by RNA modification is a way to prevent the message from RNA in being read by the wrong recipients. Now, that's an interesting statement itself. We'll come back to it again, but I want you to raise a question as we're going through all this. Who are the wrong recipients? The research team has been focused on RNA modification M6A and shown that RNA can be labeled with this modification while being copied from DNA. The work has been possible thanks to a newly developed technique, TNT sequencing, which we'll get to the definition of that in a little bit and a little bit about how it works, that labels newly made RNA for purification from the entire cellular RNA pool. So you can take and label specific RNAs that happen at a specific time. The part of the newly made RNA labeled with M6A can subsequently be isolated using antibodies. The results demonstrate that an M6A positioned at an exon next to an intron increases the RNA maturation process. It actually speeds it up, while M6A within the intron slows down the maturation of RNA, figure 2, and that's, you see, M6A at this point will give you a faster cutting out of the introns. M6A in this point, for reasons which we don't know, and I don't think they know either, slows it down. The novelty of the study is that M6A has primarily been considered a label put on mature and functional RNA, and the results therefore expand our understanding of the role of RNA modifications in RNA production and function. And that's the end of that little blurb. You see, it used to be thought this was put on whenever. Now it turns out it's put on immediately after transcription from DNA. At least in time and possibly in space. It's encoded right off the bat. Now, there's an article from evolutionnews.org that talks about encryption system found in genes, and of course is drawing an interesting conclusion from that statement. You've seen the blurb. Now we're going to look at the article itself, or the meta blurb, if you like. Encryption system found in genes is the title of this, and it starts out, researchers at Aarhus University in Denmark have uncovered a new form of gene regulation that appears to be a form of encryption of genetic information. That idea was not lost on them as they pursued the analogy from encrypted message in biological processes. That, of course, is a link to the one we just looked at. RNA modifications can encrypt the RNA code and are responsible for a very sophisticated control of RNA function. They're emphasizing, of course, the modified RNA bases have a great impact on the dynamics of gene expression. So far, this is kind of standard complexity, but then, and then it goes to say that RNA functions can affect RNA modifications can affect the function of mature RNA molecules. Programmers know all about cryptology, a form of intelligent design. Encryption is necessary when you want to conceal information from people who shouldn't have access to it. And how to achieve encryption? There are many ways to achieve encryption, such as steganography, frame shifting, compression, and other techniques. But the function is to transfer a message in plain sight such that it can only be decrypted by those with the encryption key. Let's learn how the cell does it, and then they quote another paragraph that we've just seen. And then notice it talks about a message and code and unprecedented impact of RNA in modifications at all steps of the maturation process. And then it goes on, this encryption by RNA modification is a way to prevent the message of the RNA being read by the wrong recipients. And of course, that's not something you see done ordinarily. The research team is focused on the RNA modifications M6A and so forth. The results demonstrate that an M6A position at an exon next to an intron increases RNA maturation process, while M6A with the intron slows down the maturation of RNA. And the figure 2 we've already looked at. For the encryption analogy to work, we need to see the algorithm targeting desired recipients and shielding the message from others. That's evident from their statement that the M6A modification is able to prevent messages of the RNA in being read by the wrong recipients. And an encryption key, we also need to see an encryption key that can scramble the message and a reader with the key that can unscramble it. Is this a possible role for the mysterious introns? The caption for Figure 2 explains, which I'll have to read that for you now. We didn't read it before. Newly made RNA consists of functional parts, exons, and non-functional parts, introns. The introns are excised in a process called splicing to yield a mature and functional RNA molecule composed, that obviously should be, entirely of exons. The RNA modification M6A can increase or inhibit this maturation process dependent of where M6A is deposited on newly made RNA. I still haven't seen the wrong recipients that we're trying to hide this information from. I'm sorry. It has long been a mystery why gene codes for stretches called introns that are translated but then cut out afterwards. Why genes do that. Why are they there? Here's where the findings really get interesting. Introns appear to help scramble the message, fulfilling the encryption code, but they do something else. They regulate how the exons will be assembled. The paper in Cell Reports explains how it works. Here we provide a time-resolved high-resolution assessment and so forth. We've read that. In conclusion, we show that early M6A deposition specifies the fate of the transcripts regarding splicing kinetics and alternative splicing. Still haven't seen exactly who the wrong recipients are. We've heard about M6A before as a possible player in the epigenetic code. As an epigenetic marker, M6A contains its own information, the key, that affects the products of translation. A month later, we learned that M6A might have a role in alternative splicing of RNA transcripts, and we're going to find that it does, in fact, at least influence how things are spliced, which can yield many proteins from the same gene. So this is more like data compression than it is like hiding data from the wrong recipients, as far as I can tell. Depending on how the exons are rearranged. Last year, the findings were setting off a gold rush to uncover M6A's function in the epigenetic code, and we're about to see some of the results of that. Somebody struck gold. Rate of production, and just because it doesn't say that much for our purposes, it's coming into focus is not just that having the mark that directs the regulations. Previous researcher identified a protein family called M6A readers, which recognize and bind specifically to sequences marked with M6A. So you have to be able to put it on, and you have to have an enzyme complex to do it, and it has to know when to put it on. Then you have to take it, then you have to read it by having proteins that recognize it. In addition, there are FTO pathways that respond to M6A position to either include or exclude certain exons in the transcript. We're going to find out what FTO means in a little bit. We can see the encryption algorithm coming into focus. Genes imply a key to embed introns and M6A markers, and certain readers that know the key respond to this extra information. Again, who are we hiding this from? Or is this, well, we'll come back to that. Skipping on, more work can be done to pursue this idea of encryption. For instance, what is the role of intron variability? The team found that intron length and internal M6A signal are significantly associated with slow processing. But is there additional information embedded in the introns besides length? Well, obviously M6A binding sites of some kind. Design-focused research appears best poised to understand encryption algorithms and other information-rich systems. These researchers at RS, who incidentally said nothing about evolution, seem to understand that. They're accurate, by the way. The article does not even speculate as to how this system came about. Well, let's look at the article, because I'm a firm believer that you go to the original source whenever you can. Lelupi et al., A et al. In 2018, transient N6-methyladenosine transcriptome sequencing reveals a regulatory role of M6A, that's N-methyladenosine, in splicing efficiency, and it's available on the internet. The summary, which is kind of like what we would normally call an abstract, splicing efficiency varies among transcripts, and tight control of splicing kinetics is crucial for coordinated gene expression. N6-methyladenosine, 6-MA, is most abundant RNA modification. In other words, all those hundreds of other ones that you can do, this is the most common one. And is involved in regulation of RNA biogenesis and function. The impact of M6A on regulation of RNA splicing kinetics is unknown. I want you to let that sink in for a little bit. What that means is there's a lot more in here that we have no clue about. And there's a lot that we have vague clues. Maybe it does this here, maybe not. So when we make arguments about how complex the genome is, we're making minimalist arguments. Here we provide a time-resolved high-resolution assessment of M6A on nascent RNA transcripts and unveil its importance for the control of RNA splicing kinetics. We find that early co-transcriptional M6A deposition near splice junctions promotes fast splicing, while M6A modifications in introns are associated with long, slowly processed introns and alternative splicing events. It slows down and it promotes, well, let's try joining A to C instead of A to B, or maybe A to D. In conclusion, we show that early M6A deposition specifies the fate of transcripts regarding splicing kinetics and alternative splicing. So that's what it is. Well, by this time, some of you are going, what in the world is M6A? Well, there's a ribose on the bottom attached to an adenine in the middle. Some of you may actually recognize this. And right there is the methyl group that's attached to nitrogen 6 on the adenine. Adding that methyl group is what M6A production is all about. You take that off and replace it with a hydrogen and you have just ordinary adenine, and therefore ordinary adenosine, and therefore if you attach a phosphate to this and a phosphate to that, it's part of the standard RNA chain. RNA encryption. We're going to start the article itself. We're going to look at a few things. We obviously won't read the whole thing. The RNA nucleotide code is supplemented by more than 100 chemical modifications. So that part's probably true. Greatly extending the functionality and information content of RNA. It's much more complicated than you think. N6-methyladenosine is deposited by a protein complex consisting of the methyltransferase like 3 and 14. Those abbreviations, metal 3 and metal 14, are just simply, they're too lazy to say methyltransferase like. Wilms tumor 1 associating protein. Wilms tumor? Yeah, apparently it produces too much of this stuff and it was easily purified from there first. And the viralizer homolog, something that turns boys into men, I guess. KIAAI 429. So there's 4 proteins at least in this little thing that all its job is to put that methyl group onto adenine in RNA. Early studies have demonstrated that adenosine methylation frequently occurs within a subset of RRACH. What does that mean? Well, R is either adenine or guanine. So there's 2, adenine or guanine to begin with. And then there's an A, and that A is a special A. That's the one that's going to get methylated. And then there's a C, cytosine, next. And then the last one is anything but guanine. So it could be adenine, could be uracil, could be cytosine, but if you put guanine there, it doesn't work well. So now you know, one, that you have to include in certain areas, and everywhere else in the genome, you have to not include that specific site if you don't want that adenine methylated. So you see there's certain codes you just can't use. It's sort of like, you know, there's certain words in English that you just don't use. So there's a subset of RRACH consensus sites, and that explains what that was. Fat mass and obesity associated. That's what FTO is. Apparently, if you mess with this, you get animals that are obese. And alkyl B homolog 5 are M6A demethylases. So there's a project to put this methyl on in the right place. Now there's a protein that comes along and takes it off sometimes, adding dynamics to the function of M6A in RNA biogenesis. M6A is involved in a number of RNA processes, including splicing, RNA degradation, and translation. So, you know, it makes a difference in whether you've got this one little methyl group on the adenine. These pathways are mediated in part by members of the YTH domain protein family called the M6A readers, which recognize and bind specifically to sequence marked with M6A. So once you put it on, there are proteins to take it off. Well, there are proteins to put it on, there are proteins to take it off, and now there are proteins to read it, to bind to it and, I don't know, glom up the works or something, or make it easier to bind something? I'm not sure. The presence of M6A can affect the RNA structure and increase the accessibility of the adjacent RNA sequence for the heterogeneous nuclear ribonucleoproteins, HNRNPG and HNRNPC. So apparently there are at least 7 of them, because you can get all the way to G. And you know, the other stuff is just more letters. There's so much here that we run out of things to name them. And so we kind of start doing letters, alphabet soup things. With an effect on splicing. Because of the challenging nature of addressing the impact of M6A on splicing at the immature RNA level, the direct role of MCA on splicing dynamics has not been investigated so far. Here, using TNT sequence, and TNT is another alphabet soup thing. It does not stand for trinitrotoluene. Doesn't blow up anything. It's transient N6-methyladenosine transcriptome sequencing. So it has to do with, once you've created the mRNA with this stuff on it, you want to be able to find it, and you want to be able to find it in a specific timed sequence. Okay, We show, and Q TNT chase sequence, which is qualitative TNT, which we won't bother to repeat again. Pulse chase sequencing, which means you put in something to pulse mark this. You put it in and then you take it out. And you see which RNAs actually have both our marker and the 6-methyladenosine. We show that M6A modifications deposited early and co-transcriptionally near splice junctions positively affect RNA splicing sequences. So what they're going to show is that this stuff is done almost instantly after the RNA has been translated, maybe while it's still being translated. So there's a little complex that's just waiting for the RNA to come out of the DNA transcriber, and as soon as it sees it, boom, it puts on the methyl group where it needs to be. Furthermore, we show that intronic M6A deposition is connected with slow processing kinetics and alternative splicing events. Our results strongly support a scenario in where nascent M6A deposition is functionally involved in regulating splicing efficiency, which we'll say again, SE, and alternative splicing. When they're doing all this technical stuff, it's easier to just use the abbreviations. Results: TNT sequence reveals M6A deposition on newly transcribed RNA. We developed TNT sequence to identify and study M6A on nascent RNA, on RNA that's just been put out. In brief, bromouridine labeled RNA was isolated, fragmented, and purified with a bromouridine specific antibody. So they let it run for a little bit, they homogenized everything, and they put in a bromouridine antibody, and they were able to separate the bromouridine RNA from everything else. For those who are wondering, it's 5-bromouridine. That bromine substitutes for the methyl group that would ordinarily be found in thymine, and for whatever reason, apparently it's accepted into RNA. Subsequently, M6A methylated fragments were isolated using an M6A specific antibody. The labeled RNA, that is bromouridine, RNA input, and the M6 enriched RNA fragments were sequenced to identify positions of M6A on nascent RNA. They wanted to see, well, what is the stuff that just got put out? So first they label it, then they use the label to clump it down, and then they have a specific antibody to clump down further the ones that now have methyl adenosine. So they want to know, where is this methyl adenosine and what difference does it make? M6A signatures separate distinct intron classes. Using k-means clustering, we called 3 clusters of distinct sequencing, what was it? Element dynamics, I think it is. Anyway, that sequencing dynamics, experimental process representing 4,882 fast, 5,702 medium, and 2,948 slow processed introns. So they started separating them out by which introns they had. Strikingly, we find that fast processed introns show greater M6A deposit at splicing junctions. That one I remember for sure. With an overall positive relationship between M6A deposited at 5' and 3' SJ splicing junction exonic boundaries and processing efficiency. So they were more efficient that way. In contrast, slowly processed introns are associated with an increased M6A deposition within the intron, as opposed to within the exon right next to the junction. M6A is the deposition at nascent RNA predicts SED, splicing dynamics. Intron lengths and internal M6A signal are significantly associated with slow processing, separately from each other. I'm not gonna go through all of the ins and outs of that. I don't want your eyes to glaze over. Mine glazed over, and mine are better than some. Intronic M6A deposition is associated with alternative splicing, and the evidence is pretty good. The overall intronic M6A along with the intron length are significant contributors in determining alternative splicing. In contrast to M6A and SJ exonic boundaries and strong splice site consensus sequences, that is, apparently some splices are marked better than others, SJ score ensure constitutive splicing, that is, we'll take out only one exon, or only one intron when we're putting these together. So, these are ways of marking. Well, I want this one splice exon A attached to B instead of going A to C or A to D or whatever. You put your signal in the middle of the intron and it tries to skip over. Put it in the exon, it does not. Splicing factors coincide with M6A deposition. To investigate how M6A functionality in splicing is mediated, we analyzed available cross-linking, immunization, immunoprecipitation sequencing. Notice that is another abbreviation that they'll use. Data for SRSF factors with an established role in splicing. Now, SRSF factors are not defined there. They're probably defined in Xiao et al. In 2016. We find that both SRSF3 and SRSF10, so there's two different kinds. Actually, there are probably 10 different kinds, and these are just the two most prominent ones, show a high probability to have an M6A peak summit in close proximity, that is closer than 250 nucleotides, while SRSF10 shows relatively greater affinity. The average ratio of SRSF10 to SRSF3 binding clearly separates alternative and constitutive spliced introns. The most prominently along length binned introns, putting introns into classes of different lengths. This result is in agreement with the observation that alternative splicing can be antagonistically regulated by SRSF10 versus SRSF3 binding. These results suggest that M6A could play a role in shaping the final outcome of splicing through the recruitment of splicing factors with varying M6A affinities. So SRSF3 and SRSF10 at least bind differentially to the M6A, and they're helpful in figuring out where this should be. Then the QTNT chase sequence, that fancy thing that they put together earlier, identifies M6A marked fast-tracked RNAs. So this is the ones that speed things up. To separate direct M6A mediated effects on RNA processing from sequence-specific ones, we used that long thing. Here, bromouridine labeled RNA was isolated at 0 and 30 minutes chase, and M6A transcripts were isolated with an M6A specific antibody without fragmentation. The M6A levels do not significantly differ between 0 and 30-minute chase, indicating that the overall M6A modification level of transcripts remains the same for at least 45 minutes after transcription. Skipping on, nascent M6A effects are metal 3 dependent. To provide a direct link between RNA splicing kinetics and M6A deposition at SJs, we assess the splicing kinetics after metal 3 knockdown, which means getting rid of metal 3. A 60-minute chase. For introns with high M6A signal on both 5' and 3' splice junctions. We observed a decreased splicing dynamics thing upon metal 3 knockdown for approximately half the entries. And for introns with low and medium M6A signal, we've observed an increased speed of splicing element dynamics. Discussion, we identified enrichment of M6A deposition near the 5 SJs of nascent RNA transcripts, and we show that early M6A deposition is associated with distinct RNA processing kinetics. Most importantly, we can compare the processing of individual M6A positive transcripts versus their M6A negative counterparts, demonstrating that M6A directly controls splicing kinetics irrespective of the underlying transcript sequence. Now, I don't think that that means that the underlying transcript sequence is totally irrelevant, but what it means is, of course, because for one thing you have to have an M6A splicing site or a site to create M6A, but otherwise the underlying sequence is not that important. Although the longer it is, the slower the transcription. Our findings suggest that M6A serves as a labeling signal that could be recognized by the M6A reader proteins to destine methylated transcripts for specific splicing kinetics, not only the speed, but also which one you attach it to. This is in agreement with a study describing M6A methylation as a mark for selective nuclear processing, providing evidence for an M6A-dependent RNA metabolism, or at least partially M6A dependent. Our findings furthermore reveal that intronic M6A peaks are enriched in introns involved in alternative splicing. The M6A demethylase FTO, that's that fat thing that we were talking about, binds mostly to introns and mediate removal of M6A. Knockout of FTO causes alternative splicing events with a preference for exon skipping, suggesting that demethylation of mRNA transcript promotes exon inclusion under normal conditions. Taken together, these findings suggest that intronic M6A marks that are not targeted or not yet removed by FTO mediate exon skipping, while introns involved in constitutive splicing show no enrichment in the M6A signal and most probably are targets of FTO. So there's a whole bunch of stuff that's really complicated that's going on beyond the sequence, or beyond the just simply the length or the otherwise content of the sequence. In mRNA, 6MA is enriched in the consensus DRAC motif. However, not all DRAC motifs are methylated, indicating that the presence of the sequence motif alone is not enough to drive M6A deposition. FTO clip data shows, and how do you know which ones to methylate and which ones not to? FTO clip data show no significant enrichment of the DRAC motif, leading us to hypothesize that early intronic M6A deposition is mostly in the non-DRAC sequences, where FTO can detect and eventually remove the M6A marks. So maybe FTO is simply cleaning up a little overly zealous marking of the RNA as it comes out. Recently, the M6A reader YTH-DC1 was shown to recruit SRSF3 while competing away SRSF10. YTH-DC1 binds M6A sites and promote exon inclusion. So maybe that's part of your complicated mechanism. In the absence of YTH-DC1 and SRSF3, SRF10 has the availability to bind free M6A sites independently, promoting exon skipping. SRSF3 knockdown in U2OS cells has been shown to cause exon skipping events. Using de novo motif analysis, we identify 3 additional motifs sharing an SAG core, reminiscent of the SRSF binding site consensus, suggesting that M6A could be involved in recruiting splicing factors to control SE and alternative splicing. The lack of strong consensus sequences at SJs of many introns can be compensated by the presence of M6A that could eventually attract splicing factors to exert their function. Our study shows that the crucial role of M6A on SED splicing dynamics as well as on alternative splicing is position dependent. M6A position deposited in intronic regions sort transcripts to a slow track processing pathway and is associated with alternative splicing, while M6A deposited at exonic boundaries of splicing junctions sort transcripts into a fast track processing pathway and constitutive, or maybe better yet, consecutive splicing. Anyway, that's a lot. Did anybody see who the wrong recipients were? I didn't. I would have to say that it appears to me that the Evolution News article can be misleading. Not deliberately, I don't think. It does depend on the definition of encryption. We're going to come back to that. I think they fairly got the impression that they gave from the press release. It's there. But I think the article does not support the press release in regard to encryption, and therefore does not support the argument of the Evolution News article. I can't take this information and make a nice, oh, it's encryption, it's hiding information, and then letting the right people know about it. There are at least 2 definitions of encryption. One is changing data into a form that cannot be read by some, presumably unwanted readers, but can be read by others, presumably the target audience. That's probably the most common definition of encryption. The other one is, another one is putting data into a form that requires a special reader, which is roughly the same as encoding, and why I don't like to use encryption that way. And I would have to say, if you use the first definition, the most common definition, I'm having a hard time understanding how what we know of the M6A system is designed to avoid the wrong recipients from getting information. I don't see who the wrong recipients are. I thus think that the blurb was misleading, and therefore the Evolution News article is misleading. The author should have read the original article before writing his or her article. Now, this argues for going back to the original source rather than taking the popularization. We've seen that kind of thing before. I think that the article, however, if you switch to definition 2, which is just encoding, I think that the article does bring out a heretofore unappreciated complexity in RNA coding, namely, immediately post-translational modification of RNA that influences how fast and what kind of splicing happens in creating a protein coding sequence from a gene. How much of the protein and exactly what protein you're going to make. The process of gene expression, rather than being simple, is now shown to have more and more layers of coding. Don't forget, methylation is the most common one. We're just starting to understand it a little, and there are 99 at least other ones waiting to be understood. I think they said more than 100, in which case there's 100 plus. Each layer requires several proteins, both to put on and, in our case, to take off the new code, and then to read it appropriately. It's sort of like comparing a simple computer program, Hello World, or simple factoring program, with a massive operating system, or perhaps a program suite, like the one that's being used to present to you today, Keynote, or for some computers, PowerPoint. You know, that has a little gadget here, and a little gadget here, and a little gadget here, and a little gadget here. And all of them coordinated with each other. It's stretching it to say that even the simple factoring program could originate by chance and selection. How in the world could one create a massive coordinated program suite by random mutations and natural selection? Think of it. All the people at Apple or Microsoft coming in, turning on their computers, making a few keystrokes different, and then seeing whether the program works better or not. And then the ones that actually did something that was interesting, keeping those, reproducing them, and then trying it again. Is that how you program? There is so much what you could almost call gratuitous complexity in the cell. We may find out that it's not really gratuitous. It may be actually kind of helpful, kind of necessary even. We don't know how important those are. But what we do know is that it's there, it's complicated, and each complication seems to be adding a little something. And I would raise the question, when are we justified in accepting the designer hypothesis? How complicated does it have to be before you finally fold? And with that, that's my opinion. Now it's your turn. Okay. Sure.
I came in late, so I didn't see the beginning. What was the question that got these folks going down this line?
Well, the blurb seemed to indicate that there was encryption and seemed to indicate that it was useful in somehow concealing the information from the wrong recipients. I am not sure what they had in mind, and sometimes the blurb you would assume would just allow people to do this from the article itself, and that's what it sounded like they were depending on. As we went through the article, I can't find where those wrong recipients are.
A wrong recipient would be a person that's not supposed to find it, or some kind of chemical that's not supposed to wake up and ruin everything.
Yeah, or maybe a bacterium that might use the information to its own benefit. I mean, that would be a wrong recipient, obviously, if the bacterium was hostile, or maybe a virus or something like that. I mean, I can think of wrong recipients. Maybe it's that you don't want the liver cells to know what the pancreas cells are supposed to do. I mean, I can think of where you could have a wrong recipient. I'm just not clear as to why this is hiding that information from those recipients.
In order to keep the evolutionary theory moving forward.
Well, see, of course, if you have a secret society, you pretty much have to have, or a secret message, you pretty much have to have a messenger. I mean, an open message actually kind of requires a messenger too. But a secret message, that's complication on complication, and you don't really need that. But see, even if you accept it for what I understand it to be, and that is this is an extra complicated way of doing things, you have the regular code, which itself can't be explained. If you don't believe me, ask Eugene Koonin. And, so you don't have an explanation for that, and now you have this additional layer of a message where you have to put a methyl on the adenosine, and then you have to read the that, and you even have to have some of them to come around and take off the methyl from the adenosine. And it has real-time consequences. Apparently, one of them has to do with Wilms tumor, and another one has to do with being too fat. Maybe I need that one myself, but.
Just as a trivial question, though, what organism did they happen to study this in?
As I read through, I did not see what organism they were using.
It's so common, it's not that important.
So I'm guessing they used a standard model, either mouse or human or something like that.
It seems to me the discussion here can be, to a certain extent, around the definition of encryption. And millions of things can be labeled as encrypted, as they have used the term. So many things are specialized.
Right.
You can say, hey, this is encrypted for this, and this is encrypted for that. Is there a special reason why they felt anything they identified here as uniquely special for inscriptions here, like it is in something you encrypt on a computer?
Yeah, what we need is somebody who is a native Danish or German speaker, because it's the team that was used one of those two languages, and see whether in Danish encryption and encoding or extra coding are part of thought of as the same general concept, and that there may be a translational problem. The only problem I have with the translational problem is who are the wrong recipients, because that phrase I'm having a hard time pulling out what they're talking about in terms of trying to do it from a translation error hypothesis.
To me, the significant message of this study is how complicated stuff is beyond DNA. You know, we've been hollering, hey, how in the world are you ever going to get all this DNA together and so on for life? And here we open up, well, it's not new. We've known this for years, but it emphasizes a reality beyond DNA that has to be organized somewhere or other, and adds a tremendous factor to the improbability that this could just happen by chance mutations or an evolution of all kinds of errors and only a few saved. I mean, this makes the case so much, much more impossible. It's just another layer of him.
Yeah.
I shouldn't say impossible, at least improbable in the discussion of spontaneous generation of life.
Yeah. Well, it's like somebody winning the lottery 20 times.
Yeah, well.
And then the next 3 lotteries we get, the patient, the person wins the lottery 3 more times. And at what point do you give up and say, there's design here?
Yeah. It's just, I'm just wondering, isn't the scientific community going to finally give up on this thing?
I think part of what we need to demonstrate is that at least part of this is, if I can say, theologically motivated, because really what's happening is we are not going to that hypothesis. Just not. It's a giving up on science. It's a if there is a God, then he's the God of the Galapagos and we can't stand him. Something of that nature is going on because the evidence is just, I mean, this is just one more wave on top of what you said already. You know, like I say, you've already won the lottery 20 times. Now we found out it's really 24 or 26. You know, in order to do this, you have to have what? Protein to take it off. You have to protein to put it on. And actually, it's 4 proteins to put it on. They've identified at least 4 parts to that protein machine. And then you've got to have protein readers, and it looks like we have at least 2 other protein readers that we can identify, and maybe some unknown ones that we don't identify. And the more you look at it, it gets more and more complicated. You know.
Our minds tend to accept certain things, and we get in the habit of accepting them. And then it's a little easier to accept it because we're in that habit, and we need to watch that. That's a two-edged sword, of course.
But it's, you know, it's when you talk to people who are devout evolutionists, I think that's a proper phrase. You know, the answer is, well, evolution did it here, why can't it do it here? Almost as if at some point the improbabilities don't matter anymore because, well, it happened.
Mind tends to take the shape of the container you put it in. We need to watch that.
Yeah. Yes.
I have 2 or 3 observations. Is my mic on? Anyway, the authors, I don't think, ever intended to answer the question you've raised in the presentation. And they may not even care to answer because you don't always have to make your definitions clear to make your points clear. And I think there was a method in their madness. Now, this is a lay opinion. It's just, I don't know very much at all about this subject, but it seems like they wanted to highlight the fact of the complexity of the processes. That becomes very obvious. And if you use the word encryption, in Greek you have the word kryptos, which means hidden. And so they're trying to tease out some hidden aspects of what's going on in cells and so on. So it's more or less to highlight their brilliance, I think, of being able to discover some of the secrets of the cell. And so naturally encryption it's almost like a science-fi, code word that will get people to really read and respond. Now, there's a lot of facetiousness.
Right. Remember
in what I say.
the thing on the Canaanites having descendants in the city of Sidon. Once they threw in the biblical reference, it exploded onto the scene. So
right, I remember that.
The blurb may have been deliberately designed to catch eyes or clicks or whatever you want to say. In which case, we need to be aware that that is a possibility. And when you read the next blurb, the first thing you should ask is it true? Are they really doing what the blurb sounds like it says they're doing? And I think that's an important lesson. Okay, the mic's coming down.
So my question is...
Put it up. Yeah.
So my question is, we're talking about encryption versus encoding. If it's hidden to us, yeah, we go and we discover this, but what evidence is there that it's hidden within the cell? Because this may be absolutely perfectly normal language for the cell, but we're the observers from the outside who don't know, and we're discovering, so we call it encryption. I'm sure it sounds like this is very normal processes for the cell.
Yeah. But it's normal, complicated, hugely complicated processes for the cell, which does, I think, even though it isn't conventional encryption, I think it does speak to the question of design.
But again, I wouldn't use the word complicated or complex. For us, it's difficult, but for the cell, this is perfectly normal.
Yeah.
Because that's how cells work. That's how they function. It's their language. It's their environment. It's their buildings and cars and planes and whatever. It's perfectly normal for the cell. So for us to say, it's so complex, aren't we the ones who are the idiots?
Yes. But it suggests that nature or something around nature, and the designers would usually posit a mind of some kind that's doing this, is perfectly comfortable with doing things that we would call complex.
It would be kind of strange if it was very simple, wouldn't it? It would be kind of weird.
Well, actually, if you look at beaches, they're complex in one sense, but they're very simple in another. There's little grain particles that are a certain size because that's how they've been ground down to, and they lay on a beach and they lay in all kinds of complex patterns, but really it's pretty simple. Now you come along and you have somebody start writing messages in it, suddenly, even though the message is simple in terms of how much there is in the rocks, it's complex in terms of what you expect wind and wave to do. And, if you see "I love you, Jane" and across the sand, why, there's an instant presupposition, one, that there's a mind, and two, there's a mind that wants to convey a particular message, and three, there's a recipient intended whose name is Jane. And all of a sudden, you're not talking about waves and wind and sand grains anymore. You're talking about minds. And there are people who want minds to be, well, they have to confess that they're minds because they are. But they're what they call epiphenomena that don't really in some way exist. And certainly they didn't create us, and certainly we don't have any obligation to a creator, and we don't, have any reason to believe that a creator might be wiser than the rest of us. And it's that discussion of, is there an actual mind behind all of this, that really, in a way, it's the theological driver of this whole thing, or philosophical driver, if you prefer. Yes.
Yeah, it just, the article is not going to get outside the realm of empiricism or naturalism in terms of the discussion, and it doesn't have to.
Are you suggesting that the blurb tells us what they really felt, and that the article kept that hidden?
No.
Maybe you're right. Maybe these people are secretly design advocates, and they just don't want to make it quite that obvious.
Well, they do want to get published.
Yeah, that's true.
Isn't that career suicide?
It certainly can get your paper retracted in short order if you use the word creator in the wrong way, as we have found out.
Well, there's certainly information there, but information does not imply God itself. I mean, information obviously can come from created being or natural, beings of some type. Aliens are okay as an origin of information, and because they were brought about by evolution themselves somewhere, and so you're still staying within the realm of naturalism, and
yeah.
And so as long as you keep it in that level of discussion, and then things are fine. You're not going to get this. This is not going to start a path towards recognizing the existence of God, for sure.
Yeah. As a matter of fact, we even saw Richard Dawkins toying with that idea in the movie Expelled, which just is jaw-dropping when you first hear it. Well, maybe there were aliens. And then, of course, he finishes that with, but of course they would have had to have been evolved.
Yeah. Yeah, just like God was created as well. Created God, you know.
I think we have two more questions coming from here, but before it goes there, I thought this was a wow article, and I hope they carry on with their research, because it raises lots of questions and we don't know the answers to those yet.
Yes, yes, I would have to agree with you.
I can make a theological statement now. I always like to turn it in the direction of theology. I'm more comfortable with that. Looking back at John Harvey Kellogg, one of our leading pioneers in medical work and research and so on, he was led to the conclusion that there's something called vitalism. There's something in every single cell, every blood corpuscle, every breath of life we take. He also connected with the Holy Spirit, which that got him into hot water, and probably was a perversion of he went way too far. But when you look at mechanisms like that, you've got to have traffic directors, traffic cops, you've got to have everything following certain pathways. You can't go, a lot of the operations are irreversible. I mean, it's just overwhelming when you look at the complexity, and that's what we've been talking about.
Yeah.
But I would beware of saying that this is evidence that the Holy Spirit is operating within the cell on a day-to-day basis. Now, we could debate that a long time.
Mm-hmm. No, as far as we know, those proteins are shaped in such a way that they will glom onto adenosine and put a methyl group on it, and other proteins are shaped so that once the methyl group is there, they'll glom onto the adenosine, and there are other proteins that will glom onto it and take it off, and they're designed so that they fit in certain areas. For one thing, you have to have an adenosine, but another thing is apparently in certain areas you have to have two large, purines bases in front of the adenosine, and then you have to have a cytosine after it, and then anything but guanine after that. And so there's all these parameters that have to be fulfilled before this complicated machinery will even start to work. Mm-hmm. So it's it looks like all of this stuff is mechanically done. Mm-hmm. Now, admiring a watch doesn't make you think, as you're looking at it, that the watch actually made itself, or that there's some spooky watch magic inside of the watch that has that happen. But it does kind of say somewhere along the line back there was somebody who said, I wanted time. And
if we concede that there is encryption, if that's proper to describe it, philosophically we can also say there must be an encryptor. You can carry that in a new direction.
Yeah.
If there's encryption, there's an encryptor, whether it's a material force or an individual with mind or whatever it is, but there's an encryptor. Does that make sense?
And it's arguable that from our experience outside of life, it all goes back to a mind. Yeah. Life is disputed. And I think that the people who argue that it should be a mind of some kind really kind of have the upper hand. And what's driving the other people is really more of a theological or anti-theological, if you prefer, bias. That whatever it is, they don't want God, and that kind of leaves them with a difficulty accepting the most natural explanation.
Yeah, along that, you mentioned Kellogg and his comments. Helen White makes similar ones too. And here's one where she wrote, "The God of heaven is constantly at work. It is by his power that vegetation is caused to flourish, that every leaf appears and every flower blooms. It is not as a result of a mechanism that once set in motion continues to work, that the pulse beats and the breath follows breath. In God we live and move." So,
and she made that, excuse me for interrupting, she made this statement in light of the Kellogg controversy. That's when it was dated, the 1890s.
Yeah, that's 1882 when she wrote that.
Ooh, that early.
Yeah,
No.
We're talking about a mechanism here that continues to go on by itself. So I'm wondering about the apparent inconsistency.
Well, actually, it's an interesting question. Most of chemistry and physics appears to be mechanism driven. Now, maybe that's because we're blinding our eyes to the possibility of the direct intervention of God. On the other hand, the mechanisms sure look like they're mechanics. The one place that I think that you can say that mechanics really don't fit science well is quantum mechanics. Because if there are mechanics, they're ones that violate what appeared to be the known laws of nature. I'm specifically referring to quantum entanglement and the and things like the Kocher-Specken theorem, if I remember, Kochen-Specker, I think it is. Anyway, it's one that suggests that quantum mechanics has not made up its mind until the very end. And yet it somehow obeys the laws that govern quantum mechanics. That is to say, quantum mechanics is mathematical. It is not mechanical. How do you get two particles that are far enough apart so light cannot reach from one particle to the other to agree on which way they should flip? You have to have some power that can exceed the speed of light.
No, I don't follow you on that because there's no information that's shared greater than the speed of light.
Well,
that's the issue. We don't want to get into that.
Yeah, well, maybe we should get into that sometime because I think it's an important point. Either these things are all completely mechanical or they aren't. The quote you read seems to indicate that they're not completely mechanical. And that, therefore, there is a sense in which God runs everything. And I think that God does it in an orderly way. And sometimes we can kind of understand the mechanics in terms of Newtonian mechanics pretty easily. Certainly the binding of a lot of this stuff is kind of standard chemistry and physics, although you do kind of wonder how in the world hydrogen bonds actually work, unless the quantum mechanics is really true. And one of the things I discovered out, having learned chemistry pretty well when I was young, and then going back to teaching my kids chemistry while they were taking it in college, or tutoring my kids to be precise, is that chemistry pays a lot more attention to quantum mechanics now than it used to. And there are a lot of reactions that take place that are best understood from a quantum mechanical perspective. And so maybe chemistry is more dependent on a power that is beyond what you can expect from nature alone, than we realize. But that's an interesting theological and perspective. Not so much well, some of it's a scientific perspective. We could test to see whether there's any kind of reasonable mechanics that can explain quantum mechanics, and we may wind up with that's where God is actually active in everyday life.
Just to put
comment there, and we'll come back. If
try
you
to
need.
put things in, well, a reminder of and give perspective to our discussion a little bit. For me, to remember that God's mind is light years beyond ours. We can never, ever comprehend the workings of God more than about this much. And so if he is micromanaging and we don't see it, that is no surprise to me. No surprise at all. And it's, his ways are past finding out.
Yeah. No, for what it's worth, the four laws of Newton. The first three are kind of understandable. For every action, there's an equal and opposite reaction. That kind of makes sense. Force is equal to mass times acceleration. That also kind of makes sense. The harder you pull, the faster it moves. The heavier it is, the slower it moves, And the one that threw his contemporaries, completely threw them, was gravitation, because it appeared to require instantaneous action of every molecule on Earth to the sun, and vice versa, which just didn't make sense. Mathematically, it made sense. Mechanically, how do you get the sun to pull the Earth and vice versa? And now that everybody's gone through all of the mathematics, we realize that actually, even though he wasn't exactly correct in all of the mathematics, he was to an extremely high degree correct in the mathematics. I mean, very, very close. And beyond that, we're still having the question of, how do you get it to happen instantaneously?
Well, gravitons travel at the speed of light.
Well, but you actually need something traveling faster than the speed of light to make it work, because if they go too slow, then what happens is that the graviton pulls you to where the sun was 8 minutes ago instead of now. And
yeah, well, I mean, that's been verified with the LIGOs. I mean, the latest discovery of merging neutron stars have shown that they verified that the reception of the gravitons, the pulses from the gravity waves, arrived identically with the light waves from the neutron stars.
Is this an explanation as to why the neutron stars spiral into each other then?
Well, it's the same mechanism. It's that mechanism where they spiral into each other that produces the gravity waves that they detected.
No, the gravity waves we found. That's my understanding is that there's some fairly sensitive instruments, actually very sensitive instruments that can actually detect these things.
Yeah.
But the question I have is not that there are gravity waves. It is, why should they spiral into each other? Why should they not spiral slightly away from each other if it's all done by gravitons? That, you see, you're attracted to where the sun was 8 minutes ago. The sun is attracted to where the Earth was 8 minutes ago. And if they're moving relative to each other, then then you're always going behind, and you should be going outward slightly instead of inward.
Paul, I love science. I rely heavily on science. And then I come to some very basic questions. If I follow my science, then I come to the question, does matter? Yes, I agree that probably matter exists. And then I come to the question, where did matter come from? From nothing, I'm stymied. And I have to say, there's a reality beyond science.
Well, see, this is the one thing about intelligent design that really sticks in the craw of science. Science could tolerate the Big Bang as a supernatural event. Science could live with deism if it had to. What bothers science is that it appears that life originated some, 10 billion years after 11, even if it's 1 billion, it's well after the universe got started. And yet, the claim of intelligent design is that it is complex and specified enough that explaining it on the basis of random events really doesn't help you any. In fact, it predicts things you wouldn't, that we find to be untrue now. And it looks like there was some kind of mind that started life as well as the universe. And now you're talking about something that intervenes, does things differently, however you want to explain it theologically, that doesn't fit with naturalistic science. And that's, I think, the reason why intelligent design is fighting words. And why they go berserk when somebody gets a paper that mentions a creator. I am still astounded by the fact that when Wickramasinghe and all his colleagues wrote that thing on octopi, as well as the Cambrian explosion, which is probably the more important part of their paper, actually, suggesting that this all comes from outer space somewhere, from some previous intelligent civilization. And then they get down in the middle of it and they say, well, some of us think it's God. And they managed to get that paper in. I just, it's kind of amazing, but I guess what it illustrates is that the wall that's attempted to be erected between science and God is just not really viable, and there are people who are willing to say that and other people who are willing to let them. That's the easiest conclusion I can come to on that.
Yeah, I have no problems with the mechanistic concept here, because I think you have to have an amoral nature in terms of its processes in order for free will to be a viable description as well. And
or at least a consistent nature.
Yeah. So,
and one that will allow you to do immoral things if you want to.
Yes, but nature can't determine that for you.
Yeah. If it does, then you are an automaton outside, whether you are inside or not.
Yeah. Exactly.
Oh, apparently people want to go, but anyway. It seems to me that our biggest problem is that we, as individuals, don't even know what we collectively know, and then individually and collectively we don't know enough. I mean, the thing that has freaked me out in the last 6 months is a friend or an acquaintance who is in communication on a daily basis with the ascended masters. And you say, that's all freaky, it's all corrupt, it's total nonsense. I'm telling you, you need to go near a person who's having these communications, and you realize that there is a reality we have not yet experienced, and we can hardly read about it. When you know that there's an ascended master next door to you, I tell you, you freak out. And the freaking out is based on reality which we cannot describe, and our next door neighbor will never believe, but feels you ought to be locked up in a mental hospital. And yet it is very much reality.
Anyway, come back next week and we'll be looking at the eye.